There we go. Okay, so last time we learned how an actual nerve, a motor nerve, will stimulate our muscle tissue to cause a contraction. And we learned how we generated an EPP, that end plate potential, and how we were able to then cause that end plate potential as long as the stimulus was strong enough, and we could increase, all right, our membrane potential. When I say increase, where we could make it more positive to the threshold value. Remember, the resting membrane potential for a skeletal muscle is at negative 90 millivolts. Resting membrane potential for a nerve or neuron, excuse me, is negative 70 millivolts. And then we had threshold values for both of those. It was going to be, does anyone remember for the neuron what the threshold value was? Yeah, negative 55. And so the skeletal muscle was negative 65. So we had to reach that threshold value. And when we did, right, as long as there was a sodium voltage gate nearby, we would trigger the opening of that. And then just like we saw in the neuron, we were going to see a sequential opening of those voltage gated sodium channels to open up, along with the openings, the sequential openings, followed by the sequential openings of the potassium, the voltage gated potassium channels. So we have to generate a motor action potential. And then that, I shouldn't say motor, that muscle action potential would then travel across the sarcolemma and engage or find those T-tubules, travel down to those T-tubules, and eventually trigger the opening of those voltage sensitive calcium channels. Those voltage sensitive calcium channels would then trigger the opening of those calcium release channels found on the sarcoplasmic reticulum terminal cisternum. And it would release the calcium out of those calcium reservoirs into the sarcoplasm. And then that calcium would then bind onto those regulatory proteins, the troponin, which is attached onto the tropomyosin, which was that rope-like protein that would then allow, all right, the exposure of the myosin binding sites on actin. All this should be reviewed. You should all kind of be familiar with this. And then we could get the final aspect of that muscle contraction process, which would get that cross bridge. Get that cross bridge formation, followed by the power stroke, followed by the resetting of the myosin head, all right? And then we can either cause relaxation of the muscle or another muscle contraction. So again, this should all be reviewed. So it's a good idea that you know this process. Watch that video that I've sent out to you, all right? And we'll probably watch it again after class for one more time. You know, three's the charm. But now I want to get into, we briefly talked about the metabolism. Right? These are highly metabolic tissues. Muscle tissue is highly metabolic, which means it's doing a lot. And so it has high energy demands. So now we're going to learn how we supply energy to this tissue here. And so when we're looking at our muscle cells, you're going to see here that these muscle cells have a small amount of ATP. Very little. I mean, look, 5 seconds. That's not a long time. But now some of you have probably noticed that you're able to contract your skeletal muscles for a longer period than 5 seconds. So we're going to talk about how we supply energy for that sustained contraction. So we're going to go through how we utilize the stored ATP, and then we're going to talk about how we're going to make additional ATP. So in order to talk about that, we need to talk about this very special enzyme called myokinase. So when you see myo, you should be thinking of muscle. Kinase is an enzyme because anytime you see ASE at the end of a word, you should be thinking an enzyme. And a kinase enzyme is an enzyme that likes to take phosphate and plop it onto things. It'll attach phosphate to something. And so we're going to see that in our muscle cells. You're going to have, because what happens to ATP when you use it for energy? You break it down into ADP, which is 2 phosphate molecules that are attached onto the adenosine molecule, and then a free-floating phosphate molecule. So what myokinase does is that it's just going to grab a free-floating phosphate molecule and plop it and stick it back onto ADP to make more ATP. So that's one way. So I'll show you a quick picture. We'll come back. I knew I had it in here. So if you look on the left here, you're going to see, here's our myokinase. So we have two ADP molecules here that are just floating around. And so what this myokinase is going to do is it's going to yank off, in this situation, a phosphate molecule off of this one ADP molecule, and it's going to attach it to the other ADP molecule. So, for example, our molecule on the right side here, this guy right here, we're going to take this phosphate molecule, we're going to rip it off this molecule and put it onto this ADP molecule. And as a result of that, this molecule is going to have 3 phosphates, hence ATP. And then this molecule on our right side, this ADP molecule that we pulled the second phosphate molecule off of, we're going to reduce that down to AMP, okay? Adenosine monophosphate, meaning it only has 1 phosphate molecule. So that's one of the ways that we're going to be able to recycle or make more ATP. All right, that's all fine and dandy, but we need to have some other methods to make ATP, and you'll see 3 listed there. We have this molecule called creatine phosphate, and then we have glycolysis, which is the actual, when we break down a glucose molecule into pyruvate, and that whole process gives us 2 ATP. And then we have aerobic cellular respiration. When you see the word aerobic, what are you thinking? You're thinking 1980s leggings and bad haircuts? No, I mean, you're thinking oxygen, right? I hope you are, right? Oxygen. So aerobic cellular respiration means we're going to engage in a metabolic process that includes oxygen. All right, so let's talk about these 3 different additional ways to generate ATP. Creatine phosphate. Is anyone familiar with creatine? Ever heard of it? Right? A lot of people that work out in gyms will use it, right, to help them with their workouts. And some people will say, well, it helps me recover faster from if I just did an exercise on the bench press or bicep curls or whatever exercise they're doing. It helps them to recover in between what they call their sets, their periods of exercise. And we're going to see why it does that. And there's actually some evidence out there they're starting to see that creatine is supposed to help with mental cognition, decreasing in dementia, but more, stay tuned on that. There's more out there that we're going to see, so don't go telling everybody about that yet. We're still working on that. All right, so here's what we're going to see. Much what I showed you here on this slide, right, with the myokinase, we're going to see a very similar type of scenario here with the creatine. All right, so first what we're going to see here is that creatine phosphate is only in our skeletal muscle tissue. Unfortunately, it's not in our cardiac muscle tissue, and unfortunately, it's not in our smooth muscle tissue, right? But it's in skeletal muscle tissue. But this molecule here, right, this creatine phosphate, think of it like a donor, blood donor, kidney donor, whatever you want to call it, all right? But it has a phosphate molecule attached to it. So, of course, we're going to need an enzyme of some sort. We'll make it real easy. We'll just name it after what we're dealing with here. So we have another enzyme with the kinase. So we're going to call that creatine kinase. And so again, that kinase enzyme is going to plop a phosphate molecule onto another molecule. So that's what we're going to see here. We're going to start our chemical process here, our metabolic process, with one ADP molecule and then our creatine phosphate molecule. And so this enzyme here, that creatine kinase, is going to take the phosphate off of the creatine molecule, and it's going to stick it onto this ADP molecule, giving us ATP. A lot of arrows there. I'll show you in a picture, and I'll come right back to this picture. There you go. You can see it right here on the top right side of our picture here. Here's our ADP molecule, and here's our creatine phosphate molecule. And so that's what this enzyme here, that creatine kinase takes that phosphate molecule off the creatine and it puts it onto the ADP molecule and gives us ATP. Similar to what we saw myokinase doing. It's going to take that phosphate off of one, stick it on another. We're going to wind up with one molecule that has an additional phosphate, which is going to be the ATP, and then the other molecule is going to be minus a phosphate molecule. And in this case, it's creatine. So you can see the value. Let me get rid of all this chicken scratch. You can see the value of why some athletes, why some bodybuilders, whoever, takes this stuff, because it helps them regenerate more energy for their workouts, right? And it's been very, very helpful here. So again, it won't last forever, but it will give an additional 10 to 15 seconds, which is very helpful if you're doing a sprint, like a 40-yard sprint, maybe even a 100-meter sprint. That will be very, very helpful. And so, of course, when you're done exercising, we're going to reverse this whole process. So let me come back to this slide here. So when you're done exercising, we're going to just start to take phosphate and put it onto this creatine here and make our creatine phosphate molecule again. But that's when you're resting. We'll talk about that here in a second. Does that make sense, y'all? Not too bad. All right, this picture really helps to kind of put both of these techniques or these methods into perspective here, right? And we call this the phosphate transfer system, right? So the phosphate transfer system here, because that's essentially what we're doing in both cases. So when you see kinase, think, oh, that's an enzyme that likes to transfer phosphate from one molecule to another, right? And you're going to see that again in Bio 211 when we start to talk about the metabolism of the heart. So just kind of make a little bit of a mental note there. Okay, the second way that we are going to create ATP in our skeletal muscle is called glycolysis. Some of you might be familiar with this if you've taken a biochemistry class, right? So the nice thing about glycolysis is that it's anaerobic, which means we don't need oxygen. So we can just say, bye-bye oxygen, who needs you? Right? And so what we're going to do is essentially, we're going to break glucose down into pyruvate, which is it's an intermediate molecule. I'm not going to ask you anything in addition to that, but here's the thing. If oxygen is present, then pyruvate is allowed to enter into the mitochondria. And as we know, what's the nickname of the mitochondria? Powerhouse of the cell, right? So that mitochondria, oxygen has to be present, can then break down pyruvate into additional ATP. So if you have oxygen and you have one glucose molecule, you can make 32 ATP roughly. That's pretty good. If you don't have oxygen, then and you have one glucose molecule, you can only get 2 ATP out of that. That kind of sucks by comparison, right? So it's very nice to have oxygen so we can utilize the mitochondria. Think of it like your friend that can get you into the VIP lounge at a club. All right, oxygen gets you in, okay? Without oxygen, you're not getting in. So oxygen can't get into the mitochondria, or excuse me, pyruvate can't get into the mitochondria unless oxygen is present. All right, so glucose, one glucose molecule makes 2 ATP. And this occurs outside of the mitochondria in the cytosol. So if you have oxygen, glycolysis happens first. We make pyruvate, we get the 2 ATP in the cytosol, and then oxygen helps to shuttle that pyruvate or those pyruvate molecules into the mitochondria so we can then engage in what we call the TCA cycle, also known as the Krebs cycle, or the citric acid cycle. There's lots of names for it. That's something that you'll learn in more detail in 211. If you want to learn about it after this semester's over, read chapter 3. Okay, we don't cover that. We'll cover that in 211. Okay, so that's the second way, right? Bless you, where we can get ATP. The third way is the best way. Okay, you get the most bang for your buck. Only problem is, not that I really consider it a problem, you need oxygen for this to happen. Right? So if you have oxygen and you have mitochondria, and all you need is that one glucose molecule, then what will happen is, right, that oxygen allows that pyruvate molecule to enter into the mitochondria. Now, all this other stuff that you're seeing here, we'll go into it in much more detail about how pyruvate is oxidized to carbon dioxide. Hence, you can see why in metabolically active tissues, why carbon dioxide, that's a waste, that's a respiratory waste product. And so when the cells produce the carbon dioxide, they'll get rid of the carbon dioxide by sending it into the blood, and then it gets transported back to the lungs and you exhale it. You get to learn about that all in chapter 3, right? But again, that's in 211. All right, but here's what you're going to see. All right, so we're going to see essentially a transfer of this energy through these bonds, right, to these coenzyme molecules called NADH and then FADH2. Only thing you need to really know about that is these molecules, right, especially with the hydrogen here at the end, they're going to go to what we call the electron transport chain. And that's at the very end of all of this metabolism. Again, I'm not going to test you on this, but I figured I'd tell you now because you're going to learn about it later. Right? That those hydrogen molecules, right, we are going to utilize them, right, to actually make ATP. They're going to be able to generate that energy-rich bond on the phosphate molecule. And the nice thing about this whole thing is that the waste product for this process is water. How nice is that? I wish our cars would do that, okay? So the waste product for this electron transport chain mechanism here, that this chemical bond, this transfer of these chemical bond energies to make ATP, right, our waste product is going to be water. So aerobic cellular respiration is going to yield us 30 ATP. Well, we got 2 ATP from glycolysis, and now we got 30 from the aerobic cellular respiration. That's a total of 32 ATP if oxygen is present. So you can see how nice it is to have our oxygen for these types of scenarios here. So one of the most generous molecules in your body for fuel sources or energy production are triglycerides, fats. And so even though a lot of the tissues in your body love glucose, they prefer glucose, like your brain. That's why your brain gets top billing, right? Your brain's preferred fuel source is glucose, but you don't get as much bang for your buck out of glucose. Triglycerides, you get more bang for your buck here. And so the longer the triglyceride fatty acid chains are, because you all remember a triglyceride has a glycerol backbone and then 3 fatty acid kind of tails that come off. And there's different types of fatty acids. There's short chain fatty acid, medium chain fatty acids, and long chain fatty acids. The longer the fatty acid, the more energy you can get from it. Okay, so long chain give the most. Short chain give the least here, but keep that in mind. It is the preferred fuel source. This is why if I ever went on Survivor, I would get fat. I would be pouring. I would eat glucose-rich, I'd eat carb meals all the time. I'd be 40 pounds heavier because when you go into starvation mode, right, guess what my body's going to be doing? It's going to be cannibalizing my adipose tissue and it's going to use it while all these skinny people are weak and tired. I'm going to be strong and I'm going to win it. And you guys can vote for me on the oh wait, you can't vote. You can't do that on that show. It's a different show. Big Brother, I think you can vote people out of the house. Anyways, so this slide here, figure 1017, this shows you the 3 ways, right? Or actually more than 3 ways. It shows you the 2 phosphate or transfer methods here, the myokinase and the creatine kinase. And this is more for short term. All right, and then you can see we've got our glycolysis here. So you got glucose, which is going to yield 2 ATP when it generates pyruvate. And then you've got your aerobic cellular respiration, utilizing, right, the mitochondria. Oxygen needs to be present, and then we can metabolize glucose and other fuel sources. For example, amino acids are a great fuel source, acetyl-CoA. You said we can get it for you, all right? Fatty acids, all sorts that will help to generate ATP. But again, we need oxygen to utilize aerobic cellular respiration there. All right, so what happens in a certain situation when you don't have oxygen? Right, so you're going to see here how we create lactic acid or lactate. If you've ever talked to anybody that's, or you yourself may have experienced this, like say you went for a really long walk and you have not conditioned yourself for this long walk. Maybe you're used to walking a mile. And one day you decided, I'm going to walk 5 miles. Or you just get into it. You don't realize how far you walked and you go 5-10 miles. And then the next day you're very, very sore. Right. And the reason why is you get this lactate. And this lactate will accumulate into your tissues. And essentially what a lactate is if you don't have, remember, I'm going to go back one slide here. If you look here, you got glucose, which is going to yield you pyruvate. And if you don't have oxygen present, we can't engage in aerobic cellular respiration. So what happens to pyruvate? Well, here's what happens. Pyruvate is going to then turn into lactate. Why? Virtually no oxygen or very little oxygen is available. So your body can work with lactate. It's actually pretty interesting because lactate can be used as a fuel source, right, by your skeletal muscles. And it can also be utilized, right, by your cardiac muscle and your liver. But here's what we're going to do. We're going to ship the lactate, all right, back to your liver, and your cardiac muscle cells can actually utilize it. And it's going to enter into what we call the lactic acid cycle. And so essentially what we're going to do is we're going to convert that lactate back into glucose through that process of, you guessed it, gluconeogenesis. Remember gluconeogenesis, our friend from chapter 2, right? Gluco meaning glucose, neo meaning new, right? Genesis is to create or make. So we're going to essentially, the actual definition for gluconeogenesis is the creation of glucose from a non-carbohydrate source. Because glucose is a carbohydrate, glycogen is a carbohydrate. So we're going to make glucose from something that's not carb. And so this is one of those things, all right, or items that we're going to make glucose with. So it's a nice kind of recycling. I told you, your body's great at recycling stuff. It does a really good job at it, right? Sometimes it just needs a little help. And in this situation here, you have that enzyme, that lactate dehydrogenase, which is going to help, all right, create lactate. We'll ship it off to the liver. The liver is going to throw it through the gluconeogenesis process, through the lactic acid cycle, and voila, we got glucose again. So now we can ship that glucose off to metabolically active tissue, skeletal muscle, for example, or some other tissue, and hopefully we can yield yield more ATP than what we started off with the last time, if oxygen is present. All right, so essentially when we're looking at certain activities, and so the book likes to use these examples here, I'll come back to that. You know, the 50-meter sprint, the 400-meter sprint, and then the 1,500-meter sprint, right, which is like the mile run, right? So those different activities are going to utilize certain metabolic processes that we just talked about. So for example, the 50-meter sprint, pretty short, doesn't last very long, right? That's less than 10 seconds. So we're primarily going to use that phosphate transfer system, the myokinase and the creatine kinase, right? Because it's not going to last very long. 400-meter sprint, that's once around a track. That's about a minute, okay? We're going to initially obviously use our phosphate transfer system. When we burn through all that, then we're going to fall back onto glycolysis, right? So we have those two systems that we can utilize. And then finally, we have our 1,500-meter run, okay? Anywhere between 5 to 6 minutes. We're going to use all three systems. But ideally, we want to use the aerobic cellular respiration system because you get the most bang for your buck. The most bang for your buck in that. Right? So for short bursts of energy, primarily the phosphate transfer system. Something that's going to be sustained a little bit longer, right? Then we're going to use the phosphate transfer system and then the glycolysis. And then for a long, drawn-out activity, then we're going to use all three. But primarily, we're going to kind of utilize the aerobic cellular respiration. That's why if you've ever run track or if you've ever done cross country, the coaches usually, at least some of the coaches that I know, talk to their athletes about breathing techniques. You have to utilize proper breathing because why? You're going to, inhale oxygen. You want to get as much into your body as possible, right? So you have plenty for the aerobic cellular respiration process there. All right, thoughts, questions, concerns? Well, BKB. All right, let's talk about aerobic exercise. I want to run for a mile, okay? So that type of sustained exercise is going to do a couple things. It's going to increase my heart rate, it's going to increase my respiration rate, and it's going to increase not only my respiration rate, but most likely my depth of respiration, how deep I take a breath in and exhale. All of these things that I just said to you are results of supply and demand. Your tissues demand things, and so it's your body's obligation to supply them. So, for example, that skeletal muscle, when you start to run, right, needs more energy. It starts to burn through its stored energy sources. So where can we get a lot of those energy nutrients? From your blood. So the heart is going to increase the heart rate to pump more blood to the tissues so we can get more nutrients like glucose out of your blood. But also, what else is in your blood? Oxygen. Okay? And so your lungs are going to breathe deeper and increase the respiration rate so we can pour more oxygen into your blood so it can transport that oxygen via the hemoglobin molecule in your red blood cells to the metabolically active skeletal muscle cells or whatever other cells need that oxygen. And so we're going to deliver those additional molecules to those tissues so we can sustain, hopefully, that aerobic cellular respiration there. Right? So all of that so we can enhance that oxygen delivery there. And by doing that, you can exercise longer and possibly exercise harder. Right, so that's going to be how we're going to essentially supply our tissues, that skeletal muscle tissue, with the nutrients that it needs and the respiratory gases that it needs. All right, so let's check this question out up here. Additional ATP is made immediately available in muscle tissue through which unique phosphate-containing molecule. Anyone want to take a stab at that? What do you think it is? It rhymes with biotin. Yes, you got it. Creatine phosphate. Yep. Okay, good. What are the various means for making ATP available in a 1,500-meter race? Which is the first one? Take a guess. Phosphorus transfer system. What's the second one? Glycolysis. What's the third one? Aerobic cellular respiration. Boom, there you go. Okay, so those three things are going to be, utilized during a 1,500-meter race. Okay, so that brings me to this term here, oxygen debt. Right, the definition of oxygen debt is right up there on the board, the amount of additional oxygen needed after exercise to restore pre-exercise conditions. So in layman's terms, you used a bunch of oxygen that were in these cells, right? And we need to replenish it. We need to replace it. Right? It's like if you drank all the milk in the refrigerator, you need to put more milk in the refrigerator. So that's what we're going to see here. So where do we get this oxygen from? Well, we got it from the muscle tissue, got it from this protein molecule called myoglobin. And that's the molecule that we learned about last class that stores oxygen inside of our skeletal muscle cells. Well, we have a similar molecule that wanders around in our bloodstream, and we're all familiar with this, hemoglobin. Okay, so we got to stick oxygen back onto that too. Then we used up all of our energy stores in our cytosol. So what is our main energy store for glucose? Glycogen. So we need to replace that. And then we actually have ATP molecules that we broke down in the cytosol, so we need to replace those. So of course, that myokinase and that creatine kinase are going to go to work, and they're going to rebuild ATP molecules from ADP molecules and creatine phosphate molecules. Right, but our main goal is to replenish that. And then if we have any lactates or lactic acid that we've generated, then we're going to ship that back to the liver, cardiac muscle tissue. So we're going to recycle that lactate into glucose, convert it back into glucose through gluconeogenesis. Makes sense, right? And this is all while you're resting. So when you've gone and done like some sort of exercise and you've depleted, right, oxygen and energy stores, you're going to go into oxygen debt. And we got to replace what you used. All right. I'm going to kind of move through this part here because most classes find this part pretty boring. All right. So we're going to talk about the different types of muscle fibers that you have in your body. All right. So when we classify your muscle fiber types, there's two ways that we classify them. One, what type of contraction? And two, how does it get its ATP? Right. So when we talk about the contraction, we're going to look at three aspects of contraction: how powerful, how much force we can generate, how quickly we can generate that contraction, and for how long. Okay, so power, speed, and duration. All right, so basically when we talk about power, the more the merrier, meaning the more muscle tissue you have, the more powerful a contraction you can generate. So we measure that by the diameter of the muscle fiber. So basically, smaller muscle fibers generate less power. Larger muscle fibers generate more power. And then speed and duration, all right, that relies on this molecule here called myosin ATPase. Well, that should look familiar. Remember when we were talking about ATP? When it binds onto the myosin head, it gets phosphorylated, excuse me, not phosphorylated, excuse me. It gets hydrolyzed, which is the type of chemical reaction in which we break it down into ADP and an inorganic phosphate. Well, you heard me mention the name of the enzyme is just ATPase. But now we could be specific and call it myosin ATPase because it lives on the myosin head. Remember, there's two binding sites on a myosin head. One is for ATP, and the second binding site is for actin. So we can create that cross bridge there. All right, so that's what we're going to see here, that myosin ATPase. So the type of myosin ATPase, and we'll talk about that. How fast we can actually create an action potential, and then also how fast, all right, we can release calcium from the sarcoplasmic reticulum terminal cisterna, and then finally, how quickly we can clean that mess up. All right, so we release the calcium into the sarcoplasm, all right, and then we got to clean up that party before we have another muscle contraction. So we're going to talk about how quickly we can reuptake or take up that calcium that we spilled into the cytosol and put it back into the sarcoplasmic reticulum terminal cisterna there. All right, so a lot happens. I mean, in fact, it takes a lot longer to learn this stuff than for it to actually occur. All right, so that's what we're going to see. It sounds like a lot, but that's when those four things are going to play a role in the speed and the duration of a muscle contraction. The type of myosin ATPase that we have available, how quickly we can make an action potential, and then how quickly we can release the calcium into the sarcoplasm, and how quickly we can pull it out of the sarcoplasm. All right, so there's a couple different types when we discuss our fiber types here for the type of contraction. One are, and some of you may have heard this before, fast twitch and slow twitch. All right, so fast twitch have that fast form of that myosin ATPase. So again, these types of, like the name says, fast twitch, we can get a quick muscle contraction, which means that this type of muscle fiber is going to be able to propagate and create an action potential very fast. Right? Problem is that they don't last very long. So this type of muscle fiber is going to make an explosive type of muscle contraction. So if you're going to sprint or if you're going to jump, you're going to be using the fast twitch muscle fibers primarily because they can generate a quick action potential. Right? They don't last very long, right? But they can create a lot of power, essentially. All right. So you'll see with these type of fast twitch muscle fibers, they're going to dump calcium into the sarcoplasm quickly, and they're going to take it out of the sarcoplasm just as fast. Just as fast. So everything about these muscle fibers, if you go back to this slide here, for speed and duration, everything is quick. So this enzyme here, that myosin ATPase, works quickly to hydrolyze ATP into ADP, and you all set? No worries. See you tomorrow. So the fast twitch muscle fibers, everything about it is going to be, like the name says, quick. And so if you look at what we have for the slow twitch the slow twitch muscle fibers, like the name says, they are going to be slow, all right, when we are dealing with that myosin ATPase. So that hydrolyzation process takes longer. Okay, so fast twitch, slow twitch. Now, there's one other thing that we need to mention, right, as we talk about our fiber types, and that is going to be the second way that we classify these muscle fibers is, where do they get their ATP? How do they get it? And so we have, all right, a couple ways listed up here as to how we get that ATP. The first one is called oxidative fibers, and then the second one are called glycolytic fibers. So if you look at the nicknames here for oxidative fibers, those are referred to as fatigue resistant. So those take longer to tire out. Why? Because they have more ATP on hand. So the glycolytic fibers, they're nicknamed fatigable, okay, which means that they will tire out, which means they'll burn through their energy supply fast. All right, so let's think about when we look at these terms here, because again, if you don't have to memorize, don't memorize, just try to conceptualize, try to figure it out. So oxidative fibers, you should be thinking oxygen. So that means they're going to use primarily, all right, aerobic cellular respiration to generate their ATP. So what does that mean? That means that those muscle fibers are going to have lots and lots of mitochondria, powerhouse of the cell, because that's where that aerobic cellular respiration occurs. And they're going to have lots of capillaries to bring in lots of blood, which is carrying lots of oxygen, hopefully. So when you look at these muscle fibers, they're going to have, like I said, lots of capillaries, lots of mitochondria, and we're going to have a nice supply of myoglobin. And that's that molecule that holds on to oxygen in muscles. So because of that myoglobin, right, it's going to give those muscle fibers a red coloring, just like hemoglobin does that for your blood. When hemoglobin is exposed to the air, right, it is going to grab onto oxygen. That's why if you look in your veins, that's carrying deoxygenated blood. It doesn't mean that there's no oxygen in that blood. But if you look, you can see that some of those veins might have like a blue color to them. And so, but if you were to cut that vein open, right, which you might do when you hurt yourself, if you poke yourself or whatever, the blood that comes out instantly turns red because oxygen is binding onto that hemoglobin there. So here, that's why in our oxidative muscle fibers, they'll have that red appearance there. And so these muscles can contract for longer periods of time because they have more ATP available to them. The other type, the glycolytic, I want you to think of glycolysis. Glycolysis is an anaerobic form of metabolism, meaning no oxygen, right? So that pretty much then tells me, okay, well, if these muscle fibers don't really have a lot of mitochondria because they don't have oxygen, so you'll see fewer mitochondria, fewer capillaries here, fewer everything, less myoglobin. But what we will see is lots of glycogen. Okay, because that glycogen is the stored form of glucose. And so we break glycogen down into glucose, then we metabolize the glucose to get our measly 2 ATP. But so these types of fibers, okay, don't have a lot of myoglobin in it, so they'll appear white, but they contract for shorter periods of time because they'll burn through all their ATP. All right, now that we know these different types of fibers, all right, we have our fast twitch, we have our slow twitch, and that was just for contraction types. And then here for our ATP supply, we have our oxidative and our glycolytic. Now we're going to combine some of these, and we're going to come up with a couple different forms here that I'm going to talk to you about here in a second. How does a fast twitch muscle fiber differ from a slow twitch fiber? Okay. Fast twitch have stronger contractions, quicker contractions, right? But they have shorter contraction durations. Okay, make sure you know that. All right, so now we're going to get into the 3 specific types here. How does an oxidative fiber differ from a glycolytic fiber? Okay, keep in mind, oxidative, aerobic cellular respiration, oxygen present, lots of capillaries, lots of mitochondria, right? Lots of myoglobin, so they'll appear more red. Okay, so they will have a longer sustained muscle contraction because they have more energy. Glycolytic is anaerobic, so it doesn't have a lot of that, but it does have a lot of glycogen stores, but it'll burn through that fast. So those contractions are shorter. All right. Questions about that? All right. Well, let's talk about the 3 types of skeletal muscle fibers. So now we're going to combine, all right? So we have slow oxidative fibers, which are type 1, fast oxidative fibers, which are type 2A, and then fast glycolytic fibers, which are type 2B. All right, so now we're just going to combine some of what I just talked about here. So the slow oxidative fibers, okay? Slow meaning they have the slow variant of our myosin ATPase. So therefore, these will contract slower. So slower contractions, less powerful. Here's the beautiful thing. Because they're oxidative, they've got lots of capillaries, they've got lots of mitochondria and lots of myoglobin. They've got a lot of ATP available to them. So they can sustain their contractions much longer. So you're going to see that these muscles have what we call high endurance because they have all that ATP. So these fibers, right, we can utilize. You'll see them more in your postural muscles. So for example, your erector spinae muscles will have the slow oxidative fibers. Your quadratus lumborum muscle, right, things that help to keep you upright. Several of your neck muscles that help to keep you upright, like splenius cervicis, splenius capitis, right, even trapezius. Right, some of these will have the slow oxidative fiber types, or predominantly, I should say, because you can have various types of fiber types in a single muscle. Okay, you can have all 3 types in a single muscle, right? But some fiber types will predominate depending on what that muscle does. Okay, so the second one there is the fast oxidative fiber type. So already you should be thinking, all right, oxidative. Right? That means that it's going to have a lot of ATP available, but since it's the fast variant, right, it's going to contain the fast ATP myosin, the fast variant of the myosin ATPase. So these types of contractions are going to be fast and powerful, all right, like sprinters, their quadriceps muscles will have a lot of the fast oxidative fiber types here, right? So keep that in mind. These will look a little bit lighter, like almost like a pink kind of color, all right, because they'll have, all right, myoglobin, but they won't have as much myoglobin as a slow oxidative fiber type. They'll be the reddest kind. And then finally, our last type are the fast glycolytic fibers, right? So again, these will have fast and powerful, and so you see these a lot throughout your skeletal muscle. So what type of contractions do they produce? Short, quick bursts. And so these will have, all right, they'll look almost white because they don't have a lot of myoglobin and mitochondria in them. So like I said, your muscle, your skeletal muscle will have, and it varies from individual to individual. Now, primarily what determines, take a guess, what determines what type of muscle fibers that you have predominantly in certain muscle groups? What do you think? Genetics. Genetics is primarily going to determine, right? So what you get from your mom and dad, okay, is going to determine, right, what type of muscle fibers predominate in certain major muscle tissues here, right? So again, like I said, a single muscle can have a mixture of all three of those muscle types. All right. And again, it depends on, for example, like the examples that we give up here, all right, hand muscles are going to have the fast glycolytic fibers. Why? For quickness. All right. We don't need to have the slow oxidative because they're not going to be performing sustained contractions. But you sure as heck better believe you want to have in your back muscles the slow oxidative so they can keep you upright. Right. For most of us, I don't see anybody slumped over in their chairs. All right. So your slow oxidative muscle type fibers in your back are doing what they should be doing. Right. In certain athletes, sprinters versus long distance runners, like for example, in our long distance runners, marathon runners, you're going to see the slow oxidative fiber types in their legs. But in the sprinters, you better believe you're going to see the fast glycolytic fiber types there. But keep in mind, I believe I asked a question on this. Predominantly, the fiber type is determined by your genes. And you can alter that somewhat mildly through training. Right? Somebody like myself is not going to make a very good sprinter because I don't have the muscle mass in my thighs and in my legs. It's just not in it for me. I wasn't blessed by that. I can train all I want, but I won't be Olympic caliber. Okay. So here's a nice picture. We're looking here at a muscle fascicle. So you can see the different types of fiber types here. The darker ones, those are the slow oxidative. Lots of myoglobin in there, mitochondria, lots of capillaries there. So it's going to appear darker red. And then the kind of pink fiber types that you'll see, those will be the fast oxidative. Here's one here. There's one there. Okay. Fast oxidative will be almost like a pinker, less red. And then the almost white ones, all right, those are going to be the fast glycolytic because they virtually hardly have any myoglobin, right? Not many mitochondria in there. So which muscle fiber type primarily composes muscles that maintain posture? Anyone want to shoot an answer out there? The slow oxidative. Yep, the slow oxidative ones. Long contractions. Okay. All right. Oh, good. We're getting into some cool stuff here. All right. A motor unit. So make sure that you know this because I can guarantee you I asked this on the test, or I have. And when I say the test, most likely the upcoming test, but if I don't ask it on the upcoming test, I'm going to ask it on the final. All right. So a muscle unit, know this definition: a motor neuron and all of the muscle fibers it controls. So you can see on this picture here, you can see a one single motor neuron with all its teledendria sprouting out all over the place. And at the end of the teledendria are what? The synaptic knobs. And so those synaptic knobs usually will hover or come in contact with the muscle fiber, usually in the middle of the muscle fiber. All right, but you can see this one motor neuron is going out to these various muscle fibers all over the place there. So that's when we're talking about a motor unit. A single, I'm going to keep saying this because I don't want you to miss this on the test. A single motor neuron, one single motor neuron, and all the muscle fibers it controls. So one single motor neuron can innervate one muscle fiber, or it can innervate 5, 10, 20, 30, 50, 100. Okay, right? But you have to have one single motor neuron. Keep that in mind. Right? So, what does all this mean here? Okay, I can't express this enough. I'm going to highlight it there. Single motor neuron, and all the fibers that it controls is a motor neuron. Right? So when we talk about that motor neuron, we're talking about all the various muscle fibers, cells that it innervates there. So I'm going to show you the relevance to that here. Right? And you're going to see larger muscles might have more motor neurons going to them. All right, so keep that in mind. So a motor neuron, that motor neuron cell is going to transmit the action potentials from the central nervous system, which is the brain or the spinal cord. Right? And you can see that sometimes these motor neurons will have what we call, remember the axon collaterals? Those are those little branches that come off the axon. But at the very end of the axon is the telodendrite. And that's what we're talking about, axons that branch. Those telodendrite then can innervate, all right, various, or with the synaptic knobs, can innervate various muscle fibers. Because what's a muscle fiber? It's a skeletal muscle cell, right? Make sure that you know those terms. If you don't, write it down now. Right? A muscle fiber is a muscle cell, a skeletal muscle cell. Okay, keep that in mind. All right. So when we're talking about a motor unit here, this I can't, again, I've asked this before on a test, so I'm going to underline it here for y'all. Size of the motor unit determines the degree of the control. So small motor units, all right, usually, again, this is not gospel, but less than 5 muscle fibers. So one motor neuron can innervate, all right, 5 muscle fibers. So the smaller the motor unit, the larger the precision of control. Let me repeat that. The smaller the motor unit, the larger the precision or degree of control. That's what this is. This is another one of those inverse relationships here. Okay? So the size of the motor unit and the degree of control. So like I said before, a small motor unit has a larger degree of control. A large motor unit has less of a degree of control. So in your hands, pretty dexterous, I hope. Okay, think about what surgeons do. Think about what piano players do, people that are very, very dexterous with their hands. What type of motor unit do you think goes to the muscles in your hands? Big or small? Small. Remember what I said. It's an inverse relationship. The smaller the motor unit, the larger the degree of control, which means more precision. The larger the motor unit, the smaller degree of control. Okay? So when you're talking about, the example here is the eye, okay? You're going to have small motor units there in those small muscles of your eye. So you're going to have a larger degree of control over those muscles in the eye. But think about the motor units that go to your thighs, to your legs, all right, to your quadriceps. We all know what the quadriceps are. Those are going to be larger motor units, so they will have less precision and less control. There's a reason why we do surgery with our hands and not our feet. I mean, it seems ridiculous, but think about it, all right? We have more precision with our fingers and our hands, right? A lot less with our lower extremities, although I've seen some people do some pretty amazing things with their feet, okay? But I don't want someone operating on me with their feet. Might let them walk on my back with their feet, but that's about it. All right, so that's what this picture here is showing you. Again, one more time, you can see where that neuromuscular junction is located there. like I said, and I've mentioned this before, just keep it in mind, usually the neuromuscular junction is in the mid region of the muscle fiber. What makes up that neuromuscular junction? The synaptic knob that comes from the neuron, the motor end plate that's on the muscle fiber, and then the space in between called the synaptic cleft. Those are the three things that you will have, all right, at the neuromuscular junction, which this should be reviewed because we learned about that last class. Okay, following parts: synaptic knob, synaptic cleft, and the motor end plate. Okay. Now we're going to move to another concept, okay, and we're going to talk about muscle tension, which is the power, right, that your muscle is going to generate when it contracts. So the definition that's listed up there is tension is the force generated when a muscle is stimulated to contract. So this is what I did this in college, right? We actually took a frog and did the same type of scenario here. And we measured its force of contraction. So in this situation, in this lab experiment, what we've done is we've taken the gastrocnemius muscle, which is that big muscle in a frog leg, and we hooked up some electrodes to it. And we're going to stimulate it to contract. So one, we got to make it to the threshold value. So when we determine whatever the threshold value is, and we determine that by turning up the voltage until it gives us a single contraction. And that's what we're going to do here. We're going to turn up the voltage here. Once we see a contraction, we'll see it here in our graph. Okay, so we're going to stimulate it with a certain amount of voltage there, and that's going to stimulate the muscle to contract, and then it's going to relax. And so one contraction, we call that a muscle twitch. Okay, so remember, every time a muscle contracts, what's going to follow a contraction? It's up there on the board. I'll even give you a hint. What's it do? It relaxes, yeah. So muscle contracts, followed by relax. Okay, so that's what we call a muscle twitch. So contraction, relaxation. Okay, so now we're going to see, all right, how do we manipulate that, okay, to get it to contract harder and longer. So the definition of an actual muscle twitch, you want to know this, a single brief contraction period followed by relaxation, right? And that all came about with a single stimuli. All right, we determined whatever that threshold value was with our voltage apparatus there. And so once we've achieved that, we get that minimal, that baseline, that minimum amount. We push the button, it sends off a certain current, and that current gives us a muscle twitch, contraction followed by relaxation. All right, so what else should we know when we're talking about a muscle twitch and this? You'll notice here, I'm going to go back here. You'll notice, all right, we have this latent period. We'll talk about what that is. And then remember what follows a muscle contraction. All right, it's very similar to what followed, all right, when we stimulated the nerve. Remember, we had that period of time in which we couldn't stimulate it again. And we're going to talk about that, right? That refractory period. We're going to get to that, right? But so when we're talking about the twitch, okay, that muscle twitch, we have that latent period, what we call the delay. Because we've got the stimulus there, that voltage there. So we get to that voltage, we push the button, but yet it doesn't contract immediately. And we'll discuss what causes that. It doesn't contract immediately, but it does contract though. All right, and so there's that brief period, that delay period, which we call the latent period here. And we'll talk about that. Then, of course, we have the contraction period, okay? And that's when all the power strokes occur. And then the relaxation period, where we've disengaged our cross bridges, all right? And then we start to get rid of that calcium. We suck it out of the sarcoplasm, and it goes back into the sarcoplasmic reticulum, terminal cisterna there. And so obviously the relaxation period is going to last a little bit longer than the contraction period. Hence, look at the picture there. And you can see this is more vertical. So the contraction period is more vertical, it's quicker, and then the relaxation period is more horizontal, right? And it's longer. Right? But again, You can see the latent period is this brief period of time where we initiated the stimulus until we get that contraction there. All right, so what events are occurring in a muscle that produce the different components of a muscle twitch? And there you go. Those 3 periods there, okay? The latent period that leads up to the actual contraction, when we start to generate force and tension inside of that muscle. Then we have the actual contraction, where we get the cross bridge formation, shortening of the muscle fibers there, followed by the relaxation period where we disengage those cross bridges, and then we get the actual relaxation occurring because calcium is leaving the sarcoplasm. Okay, so now what we're going to do is we're going to start, instead of tapping that button once on our voltage meter there, we're going to tap it repeatedly, and we're going to see what happens. Okay, so if we increase our voltage, we're going to recruit more motor units. And if we recruit more motor units, what does that mean? That means we're recruiting more muscle fibers. And if we increase more muscle fibers, we can increase our muscle tension. All right. So as we recruit more, this process is called multiple motor units summation. Remember, summation is to add up. We're going to add these motor units up together. So think about this. If you're going to pick up a pencil compared to you picking up your backpack, your computer bag, or whatever, which one do you think you recruit more motor units for? Picking up the pencil or picking up a heavy backpack? Yeah, of course. Okay, so you have to recruit more motor units to pick up that heavy backpack. So we're going to see now through that multiple motor unit summation, this is what's going on here. All right, how the same muscles that you use to pick up that pencil, you're using them to pick up a heavy backpack. So that must mean something, okay? Because you don't need as many motor units or as many muscle fibers to pick up the pencil as you do picking up that heavy backpack here. Right? So what we're going to talk about when we get over that minimum voltage value there, we're going to see recruitment of more muscle fibers increase until eventually we recruited all of them. So we're going to plateau, essentially. So as we increase the volume, we're going to increase the muscle fiber recruitment. Thus, we're going to generate more muscle tension, a stronger contraction, but eventually we're going to hit a plateau because we've recruited everything and we cannot recruit more. All right, no matter how much we turn that voltage up. Okay, all right, let's get back on track here. All right, so as we're going to see, as we increase the tension or the voltage, we're going to increase the recruitment of those motor units. And so initially, and this is something that you want to know, is we're going to initially recruit the smaller ones first, and then the larger ones come online later on, which makes sense, right? Because like when you go to pick up that pencil, all right, you don't need as much. You don't need to have, why isn't this going away? There we go. So that's what we're seeing here on these diagrams here. So on the left side, when we're talking about individual muscle twitches, what you're going to see is we're going to stimulate the muscle fibers. We get a contraction followed by relaxation. And then we'll just keep doing that as long as we give enough time for the relaxation to kick in. So now what we're going to do is we're going to increase the frequency. And as we increase the frequency, we're going to start to recruit more motor units. And as we increase the frequency, we increase the voltage, and you'll see our muscle contractions become stronger and stronger. And then eventually there's less time for relaxation. So it almost feels like the muscle is going into spasm. And then eventually we get one long spasm period here because we've turned the voltage up as high as we've eventually recruited all the muscle fibers. So, and as the voltage is kicked in there because we've increased the frequency, the muscles aren't getting any time to relax. The only time that they're able to relax is when they go into fatigue, when they burnt through all of their ATP, right? So that's what you just saw here. We're seeing what's called that wave summation. Each successive stimuli is building on the previous one, right? And that builds our muscle tension there, right? So we have our wave summation. And again, am I going to ask you what the frequency needs to be? No, right? But it is around 20 per second there. And so we're causing muscle contraction, but we're not giving the muscles, all right, a complete amount of relaxation there. And as we recruit more muscle fibers, our contractions become stronger. And so we see the two types of tetany. You get the incomplete tetany, which is where we get a slight amount of relaxation before the next contraction begins. And then we get what's known as just complete tetany here, or tetany, right, in which we don't get any period of relaxation. It looks like one prolonged, sustained muscle contraction there. And that's when we get the frequency of the stimuli up really high, 40 to 50. Okay, we're just stimulating that muscle. And we did that in lab when we were doing that frog leg. We cranked up the voltage there, and we cranked up the frequency, and you could see that muscle, that frog leg, just contract. It wasn't relaxing there, right? So that high frequency will eventually lead to that fatigue. Okay. So that is, all right, what happens when we change our frequency. So that's what you're kind of seeing here. As we have, all right, you can see as we increase the voltage, we start to increase the muscle tension until we have that sustained muscle contraction, or not sustained, but we can't get it any higher because we've recruited all the muscle fibers. You can see on this one, it looks, you're seeing, all right, at 0, no muscle fiber recruitment. At 1, no muscle fiber recruitment. Now, right, we turn the voltage up a little bit. Hey, we got some muscle fibers. Now let's turn that voltage up more. You can see how we're recruiting more and more muscle fibers as we increase the voltage there. And then eventually we've recruited them all. So we can't get a stronger muscle contraction because we don't have any more muscle fibers to contribute to it because we recruited all of them. All right, which brings me to muscle tone. All right, definition of muscle tone is resting tension in a muscle. So I'm sure some of you have either been a recipient or the person that actually gives, all right, somebody a back massage. And I'm not trying to sound weird on this, but you know what I mean? If you ever put your hands on the back of somebody and you feel their muscles, you're like, oh man, you're tight. That's what they're referring to. That's actually, you should be saying, hey, you know, your muscle tone is hypertonic if you really want to be specific, or you could say you're tight. But what we're seeing is, right, your nervous system. Does anyone happen to know what part of the nervous system is responsible for this? I'll give you two choices to pick from. Autonomic nervous system or somatic nervous system? Take a guess. What's responsible for muscle tone, that resting muscle tone? Do you think it's autonomic nervous system or the somatic nervous system? The other one, somatic. Because remember, the somatic nervous system only affects one type of effector, which is skeletal muscle. Autonomic nervous system affects all the other types of effectors, glands, cardiac muscle, and smooth muscle. All right. And so when we're talking about muscle tone, primarily we're talking about the resting tension in skeletal muscle tissue here. So what that basically means is the nervous system is sending out action potentials to, all right, some of the motor units in these fibers. Like you can touch your forearm, for example, right, in certain, or your neck muscles. And so some of those muscles obviously are going to be trying to sustain, all right, your upright posture, okay? But when you're laying down, your muscle tone should decrease significantly, and especially when you're sleeping. I do it all the time. I see it all the time on patients. When a patient is standing or sitting up, you can feel their back muscles. And then when they lay down, their back muscles should be relaxed because they're no longer sustaining the back. So their muscle tone should drop, right? But that's what we're seeing here with our muscle tone. Random motor units are being stimulated. Okay, that's your somatic nervous system that is just hitting up these motor units here. Okay, not to the point we don't want to fatigue them, but we just basically want to make sure that there's a good connection there, right? But when that person is in the recumbent position, when they're laying down, most of their skeletal muscles should be in a good resting tension state, but especially when they're sleeping. That's the best state, okay? Because you're not doing anything. And we'll talk about what happens during the sleep period because we're going to talk about the RAF. All right, real quick, a couple different types of contractions. This is one of your essay questions on the Blackboard discussion section here. It talks about the different types of muscle contractions. The two main types are what we call isometric muscle contractions and isotonic muscle contractions. An isometric muscle contraction is if you were trying to push against a brick wall, right? The muscle's not shortening, okay, because the wall is not moving. What does that mean? All right, the muscle stays the same length. Bodybuilders do this all the time when they're on the stage posing. They're holding a pose there, right? They are isometrically contracting their muscles. They're not shortening, okay, but they are causing, all right, muscle tension to occur. So here's what we're going to say. The definition of isometric muscle contraction is we're increasing the muscle tension, right? But it's not enough to overcome the resistance. So that example that I gave you, when you're pushing on a brick wall, right? You're not pushing that brick wall down. You're not breaking through the brick wall, right? And your muscles aren't shortening, okay? Because they're still generating, but they are increasing their muscle tension there or that force there. So that's an isometric contraction. An isotonic contraction, there's two types, right? But basically what we're seeing with an isotonic contraction, we do overcome that resistance. So we're actually going to see some sort of movement occurring here, right? But there's two types. One, we shorten the muscle. The other, we cause the muscle to lengthen, okay? So a concentric contraction, the muscle shortens, right? And I got a nice picture here we'll see on the next page. There's a picture of a guy picking his baby up and bringing the baby towards his head. So when he lifts the baby towards his face, he's contracting the biceps brachii muscle. He's flexing the elbow. So the muscle is shortening. So that is the concentric, right? The eccentric is when you lengthen the muscle, but the muscle is still contracting. So think what happens. So the dad brought the baby to his face to give it a hug or a kiss or whatever. Now he's going to put it back down in the crib. So when he starts to lower it back down in the crib, his biceps muscle is still contracting, but what's it doing? All right, the elbow is extending, all right, because the triceps muscle, the triceps brachii, that's concentrically contracting. It's shortening because we're doing extension at the elbow. But at the same time, the biceps brachii is also contracting because if it didn't, this is what the dad would do to the baby. All right, baby, I'm going to put you down in bed. You know, that's what would happen. If the only muscle that was working was the triceps. So this is where we get into agonist and antagonistic muscle contractions. So when you attempt to show, oh wait, hold on. Thought I had the picture. There we go. Here's isometric versus isotonic. You can see when the dad starts to bring the baby towards his face, he's concentrically contracting the biceps brachii. Then as he holds the baby there, that is going to be isometric. Then when he lowers the baby back down to put the baby back into the crib, the biceps brachii muscle still contracts, right? But it lengthens instead of shortening. So that brings me to this question here. When you attempt to shovel a load of snow that is too heavy, what sort of muscle contraction are you using? Now, this is kind of lost on a lot of us down here because we're in the South and it doesn't really snow here too often, right? But it's happened to me plenty of times where I've taken a shovel, stuck it into the snowbank, and I tried to lift that pile of snow up and I can't do it because it's too heavy. So if no movement's occurring, what type of muscle contraction is that? Isometric, that's right. Okay, isometric muscle contraction. I'm still contracting my muscles, no movement's occurring because I'm not able to overcome the resistance there. And that's what you see here. All right, this leads me into how, right, and we again up north, we saw it all the time where they would say, we caution people from shoveling the snow, all right, when it's wet and very heavy, and especially older folks, because isometric muscle contractions can increase blood pressure. It causes you to engage in what we call the Valsalva maneuver, which is when you increase your intra-abdominal pressure inside. All right. So when you sustain these muscle contractions, you can increase that blood pressure. If you have increased blood pressure, that's a very bad thing to have happen. And also, right, because you're in a cold environment, you're going to get what we call vasoconstriction. Now, you'll learn the purpose of this when you study the circulatory system in 211, but vasoconstriction increases what we call peripheral resistance, right? When you increase peripheral resistance, you're going to increase your blood pressure, right? You'll learn about this later on. Alrighty. I think what I'm going to do is stop right here because I've gone over a little bit and it looks like your brains are full.